The skin is the largest organ of our body. It surrounds it and protects it from dehydration and environmental insults. And so far, it has not been known how it develops during development. In this work, we found out that the skin develops in 2 phases. The current view in the field is that the switch in cell division orientation from the parallel mode to the perpendicular mode is what switches a single-layered epithelium into a stratified epithelium. But what we found is that indeed we can disrupt this cell division orientation without affecting epidermal development, indicating that the 2 processes are independent.
To assess whether there is a link between cell division orientation and epidermal stratification, we used centrosomes as a tool. And centrosomes regulate the mitotic spindle, and when we lose the centrosomes in the cells of the developing epidermis by mouse genetic models, we were actually able to see that this leads to a drastic shift in the cell division orientation, but surprisingly, this had no major effect on the epidermal stratification and differentiation. Thus, this led us to conclude actually that the 2 processes of cell division orientation as well as epidermal stratification can be independent of each other. And moreover, to understand which processes are really contributing to the epidermal stratification during development, we used time-lapse imaging of the developing mouse epidermis and could observe that both cellular delamination, which is a process where single cells leave the basal layer and move upwards, as well as cell divisions are contributing to epidermal stratification. Resulting, we concluded in a 2-phase model of epidermal stratification, where during the first and major phase, we think that this is mainly characterized by high proliferation rates, so many dividing cells in the layers of the epidermis, as well as cellular detachment of single cells from the basal layer as contributing. Moreover, during the second phase, this is more like a maintenance phase, which can be supported by delamination, but is not really linked to cell division orientation.
We were surprised to see that there was an increase in the production of the number of cells that the single-layered epithelium could not accommodate anymore, and this is pushing the cells to go up and form the new layers.
We think that this also might be a basic principle which can also be applied not only to the developing skin epidermis but also to other multi-layered epithelia, where we see that crowding can lead to cells being pushed upwards to generate multiple layers.
We found in our work is that not only is the basal layer dividing and proliferating, but also the suprabasal, the newly formed suprabasal layers. Therefore, we looked at human hyperproliferative disorders to see whether this is also a conserved mechanism between the basic developmental process that we discovered and these disorders. And we found that indeed the suprabasal layers in these hyperproliferative disorders, namely psoriasis and atopic dermatitis, had an increase in cell division in the upper layers as well. Understanding the exact switches from proliferation to differentiation in the epidermis, which is what we're trying to do with this work, can illuminate what happens during disease situations in atopic dermatitis and psoriasis, and therefore provide druggable targets to treat those diseases.